Welcome to the series. In the last lecture, we have elucidated. We have discussed about the main important hallmarks in the last discussion. Now, let us elucidate important hallmarks that are important all the hallmarks in a very detailed way. But the basic necessity for discussing each hallmark in a neat way is most of the therapeutics, and in fact, most of the targeted therapists now do target the important hallmarks, and that's how the development of the drug begins. The first hallmark, as discussed earlier, is self-sufficiency in growth signals. So cells in the body require hormones or other molecules to act as the signals for divide and to grow in resistance. The cells have ability to grow, and without these external sequence as a consequence of oncogene activation. As told earlier, every cell has capability or capacity to divide, but this capacity to divide is put in under control. So we need to remember that there is a capacity of divide that is conferred by the proto-oncogene, but proto-oncogene doesn't act because it is put under constraint. In cancer cells, this constraint is removed. So proto-oncogene transforms itself into an oncogenes. So if you see here, there are three types of them. They are chemicals, they are viruses, and they are radiation. These transform a proto-oncogene into activated oncogene. So proto-oncogene is present in a normal cell, and activated oncogene is present in a cancer cell. Now, what does this activated oncogene comes is the subject of discussion. Oncogenes will produce oncoproteins. These oncoproteins will cause excessive cellular growth by manipulating or bypassing, manipulating the growth pathways or bypassing the growth inhibitory pathways. So we will now discuss of what are different categories of oncoproteins. What are different categories of oncoproteins? The first category is the growth factors. Second factor is growth factor receptors. The third factor is signal transduction molecules. The fourth one is transcriptional factor activation. And the fifth one is cell cycle protein activation. Now let us go in details of growth factors. Usually normal cells will not synthesize their own growth factors. That is a constraint that is present on the cell to stop the proliferations. But in certain cases, cancer cells will synthesize the same growth factors for which they are living, especially platelet-derived growth factors, etc. So examples for this one is platelet-derived growth factors, usually expressed in astrocytoma. So astrocytoma, due to the conferred genetic mutations that happen in astrocytoma, lead them or transform themselves in secreting PGDF. This PGDF in turn activates its own cell, and which will lead to proliferation. Fibroblast growth factors usually secreted in osteosarcoma, stomach cancer, bladder cancer, or breast cancer. TGF alpha is also secreted in astrocytoma. Human growth factors is usually over-secreted in hepatocellular carcinoma and thyroid cancer. The next level, from the growth factor, now growth factors go on them attached to a growth factor receptor. By mutation of the growth factor receptor, making it active, or increase in the growth number of growth factors, what we do, this also causes cellular proliferation. So this is most commonly therapeutically used. Usually proto-oncogenes encode a growth factor, but when the proto-oncogene is mutated, or when there are multiple copies of proto-oncogene, the number of growth factor, activity of growth factor may be increased, or there may be many more number of growth factors. This will lead in increase in the cell survival of the cell. The most important growth factor is epidermal growth factor receptor 1. It is a transmembrane glycoprotein with an extracellular epidermal growth factor domain and intracellular tyrosine kinase domain. Sometimes the disturbance or mutation in this tyrosine kinase domain also causes intrinsic activity of the growth factor receptor. As told, usually growth factor receptor is under control, but due to mutation in the tyrosine kinase domain, the intrinsic control activity is lost. So they will be activated all the time. One example is EGFR tyrosine kinase mutation in inframe mutations of exon 19 or point mutation in exon 21 of the EGFR gene, the gene that encodes epidermal growth factor receptor. can have mutations in in-frame mutations of exon 19 or exon 20. These mutations will lead to constitutional activation of the tyrosine kinase of the epidermal growth factor receptor protein. This leads to constitutively activated epidermal growth factor receptors. So if we can find a solution where we can inhibit the tyrosine kinase, we can actually stop the activity of growth factor receptor and thus the survival of the cell. So what are the two most common drug molecules are Geftinib and erlotinib, which are the first generation EGFR inhibitors, which will inhibit the tyrosine kinase domain of the epidermal growth factor receptor. Then we have other thing, epidermal growth factor receptor 2, usually called as HER2. In this case, as shown here, normal breast cancer we usually will have, for example, a four epidermal growth factor receptor. But an abnormal HER2 positive breast cancers will express so many HER2 proteins that the cell becomes more sensitive to the growth factors that are present externally. This makes the cells to proliferate. Though the number of growth factors are less, due to amplification in the number of growth factor receptors, there is increase in the survival of the cell. Then we have ALK receptor. ALK receptor plays a pivotal role in cell communication, in normal development and function. Mutations in ALK receptor gene lead to overactive ALK receptor, especially seen in adenocarcinoma, as exemplified by crizotinib, alectinib, ceritinib, and larotrectinib, which is recently used. Then we have FLT3 inhibitors, which is usually seen in acute myeloid leukemias. These sometimes FLT3 mutations can be mutated. There can be internal tandem repeats or there will be mutations. So inhibitors of FLT3, which is called FMS-like kinase, can also decrease in the cell growth and survival. The common inhibitors of FLT3 mutations are sorafenib, sunitinib. The most specific is the midostaurin. So this is a chart which is explaining growth factor receptors, the mutations, and the common cancers that are seen. So EGFR mutation is usually seen in lung cancer and breast cancer. FMS like tyrosine kinase is seen in leukemia. Receptor for neurotropic factors are seen in point mutations in MEN1 and MEN2. PGDF receptor mutated in glioma. KIT ligand, most important, is mutated in case of GIST and seminomas and leukemias. All receptors are usually mutated in adenocarcinoma of lung and certain lymphomas, and also in most of them also neuroblastoma. All receptors are now most commonly used in relapsed refractory neuroblastomas. Then now let us follow the. There is a growth factor receptor. There is a tyrosine kinase which will influence its activation and deactivation, and then we have a signal transduction molecule which acts downstream to the receptor. It acts as a postman from the cell membrane to the nucleus. Once that receptor receives a signal, it undergoes conformational change, which in turn launches a series of biochemical reactions in the cell. These intracellular signaling properties are called signal transduction cascades, and they typically amplify the message, producing multiple intracellular signals for every one receptor. And mutations in these signal transduction molecules also contribute to cell survival and growth curve. The most important signal transduction molecules are RAS proteins. Usually RAS is HRAS, KRAS, and NRAS. They are regulators of internal signal transductions. They are membranes of small associated G proteins that bind to guanine nucleotides. So stimulation of receptor kinases by growth factor leads to exchange of GDP for GTP and subsequent conformal changes. They generate activated RAS. You see here, so just follow the diagram. I'll just put a slide here. You can follow the diagram. So this is a growth factor, and this is a growth factor receptor. Okay, and bound to the cell surface, there is called a RAS receptor, which is a signal transduction molecule, which is responsible for interfering the signals that are present. So the RAS receptor What is activation of RAS receptor is transient because it has intrinsic GTPase activate. How now remember how is the RAS activated? It RAS is activated by exchange of GDP for GTP molecules and subsequent conformal changes in that activate RAS. So inactive RAS is converted into active RAS. So RAS again activates RAS. RAS will again activate MAP-PK and MEK kinase pathway, which is seen in most of the cancer, especially RAS, RAF, MEK pathways altered in malignant melanoma. Once activated, RAS protein signals in nucleons leading to cell proliferation. So mutations in any of this pathway, either growth factor receptor, signal transduction molecules, will lead to survival of the molecules. So as told earlier, there is something called the RAS proteins are again dependent upon the GTP to GTPase. So the RAS proteins are controlled because of the GTPase molecules that are present. So mutations in these GTPase molecules are aberrant GTPase molecules also can cause activation of RAS indefinitely. So first, let us look in them into two separate things. One, RAS activating mutations. The point mutations in RAS family genes constitute the most common tumors of proto-oncogenes. So they are divided into HRAS, KRAS, and NRAS. So 50 to 20% of humans have RAS. So 90% of pancreatic cancers, 50% of colon, and 30% of lung adenocarcinomas and myeloma harbor RAS mutation. In fact, RAS is the most common cancer. But the problem is we couldn't generate any molecule which is specific for RAS protein till now. So as we told earlier, RAS protein is inactive when it is bound to GDP bound, and it is active when it is bound to GTP bound. So this is mediated by GTPase activity. When the GTPase activity is altered, RAS can be always in constitutively active form. So constitutively active GTPase activity is seen in certain kind of diseases, which will inherently predispose the cancer to form the tumor, predispose the person to form tumors. These mutated form of RAS are trapped in activated GTP bound, and as a result, cell receives no pro-growth signals continuously. Loss of function mutations in GTPase that increase the RAS activity by removing the constraint. Disabling mutations, these are disabling mutations in neurofibroma in a GTP encoding, GAP encoding by NF1 gene are associated in inherent cancer. Now, let me explain. So, there is a gene called neurofibrin 1 in the DNA of a normal person. This codes a normal GTPase activity gene which controls RAS. Sometimes what happens, this sequence of the DNA, the neurofibrin 1, is mutated. Once it is mutated, the RAS is not under normal constraint, it will be starting forming tumors. This is called as neurofibromatosis, and the gene that is responsible for neurofibromin 1. Neurofibromin is a tumor suppressor gene and acts through negative regulation of RAS. If you see here, what does neurofibromin 1 actually? It stops mutation. It stops the mutation. It stops the mutation by maintaining GTPase activity. Thus, mutation of a substance which will control RAS, which acts as a tumor suppressor gene, will in fact lead to increased formation of tumor metastasis. And as seen here, there are downstream pathways I have shown in this picture. RAS, downstream of RAS is RAF pathway. So mutations in BRAF usually seen in melanoma, hairy cell leukemia, lung cancers, and lung cancers also contribute to survival energies, and mutations in RAF protein or aberrant RAF proteins also lead to carcinogenesis. So it is basically a downstream molecule. It is upstream to MAP ERK pathway, but it is downstream to RAS. So it is usually seen 100% in hairy cell leukemia, that is in 60% in melanomas. Then we have another most important pathways, PI3 phosphatase, inositol kinase proteins family. So if you see very carefully, they are receptor tyrosine kinases. So they are downstream proteins of the RAS pathway, other than the RAF pathway. They act, it is a serine threonine kinase, including AKT. So PIK3 will activate the AKT. AKT phosphorylates more than 150 proteins and acts as a major signaling molecule. One of the major molecule among this is mTOR pathway. It's key regulator of proteins and apoptosis. mTOR pathway, which is mTOR pathway is a key regulator of protein synthesis and apoptosis. So mutations in kinases in PIK3 pathway are common in certain cancers, especially in breast cancer. Once we give endocrine therapy, some people are intrinsically sensitive to endocrine therapy due to PIK3 mutations. So there are specific PIK3 inhibitors which contribute to which are useful to control cell proliferation. They are now PIK3 inhibitors are now indicated in chronic lymphoid leukemia and breast cancer. So PIK3 can also be inactivated by P10. So PIK3 is unriddled by loss of function mutations in its negative regulator called P10. Thus, P10 gene mutation indirectly leads to activation of PIK3. It's like PIK3 is in the control of P10, and mutation in P10 will increase the activity of PIK3 mutations. So mutations in P10 is usually seen in Cowden syndrome. So these kinds are the one which usually leads to haploinsufficiency. Like decrease in the P10 levels, though the P10s need not be knocked down in the both chromosomal sets, but decrease in the P10 levels will slowly increase the activity of PIK3. And PIK3 is, as told earlier, PIK3 controls AKT. AKT is responsible for most of the things, and mTOR is also one of the downstream pathway of PIK3 mutation. So then the most common, like a few years ago, the Time magazine, not few years ago, it has already passed some 20 years. Time magazine has told the cure of cancer as a pill. So it's receptor ABL tyrosine kinase mutation. So ABL tyrosine kinase is usually expressed in the nucleus, and it is responsible for certain genetic regulation. Sometimes what happens due to a mutation, due to a chromosomal translocation from 9 to 22. So ABL, the normal ABL is converted into an normal ABL, which is a proto-oncogene, is converted into BCR ABL, which is an oncogene. So ABL proto-oncogene usually produces nuclear protein kinases that is implicated in cell differentiation, cell division, and cell adhesion. Activity of ABL protein is negatively recruited by SH3 mobile, and deletion of SH3 mobile turns ABL into an oncogene. So what happens in 9-level translocation? This lead to head-to-tail fusion of BCR and ABL gene, leading to fusion gene, leading to constitutively active ABL. So usually ABL is inactive in cytoplasm, but BCR ABL is active in cytoplasm, and thus it will stimulate cellular proliferation and all those things. So the constraint of ABL is removed with the help of by translocation of ABL molecule from chromosome 9 to chromosome 22. This will lead to overactivity of ABL, and that's lead to formation of cancer. So as told, normally ABL gene is present on the 9th chromosome, and due to disparency, the 9th chromosome ABL gene is in control by SH3 molecules when it is present in the 9th molecule. But once the ABL gene is transferred to chromosome 22, instead of being under control under ABL molecules, it is just adjacent to the BCR locus. This BCR locus and ABL locus contribute to each other, and they sustain the proliferation pathways in the body. Then we have JAK2 mutations. JAK2 is a non-receptor tyrosine kinase that participates in JAK-STAT signaling, usually responsible for platelet productions and also platelet production, red blood cell productions. So normal JAK pathway, it is bound to the erythropoietin receptor. So and it has this negative feedback regulation. So once it goes and stimulates, it automatically is regulated. But in polycythemia vera, there is a mutation in the JAK2 STAT pathway, which will lead to aberrant activation, and this leads to formation of polycythemia vera, which is like unregulated RBC production. It's a chronic myeloproliferative neoplasm. So proteins that are involved in signal translations. So it's the RAS protein, and the RAS protein, how it is inactivated, it's inactivated by GTP join. It is inactivated by the GTPase mutations also cause inactivation. So RAS proteins also can undergo point mutations. This is seen in colon cancer, KRAS, especially colon cancer, lung and pancreatic tumors, HRAS is in bladder and kidney tumors, NRAS in melanoma, immunological malignancies, and GNS and pituitary adenoma. And non-receptor tyrosine kinase ABL is mutated. the ABL is mutated due to translocation in chronic myelogenous leukemia. RAS signal transduction is mutated, so it will lead to formation of breed of protein seen in melanomas, leukemias, colon carcinoma, and others. Notch signal transduction is seen in leukemia, lymphoma, and breast carcinoma. JAK-STAT as earlier. So notch signal, what do we mean by notch signal? Notch is also an important signal transduction molecule. In fact, notch is the one which directs the cell to which kind of differentiation it's happen. So in leukemias and lymphomas, in leukemia and lymphoma, the initial event happens in the stem cells. So stem cells, instead of being directed to form one particular lineage of cells, they are mutated and form form proliferation of the stem cells instead of differentiated into normal stem cells. So this mutation can be notch mutation may be the reason why differentiation is halted and there is increased proliferation of the stem cells. Again, after signal transduction molecules, there can also transcription factors. So final consequence of deregulated mitogenic signal pathways is inoperative continuous stimulation of nuclear transcription factors that drive growth promoting genes. So transcription, you remember transcription is formation of RNA from DNA. So this transcription is also regulated by certain factors. They are called transcription factors. So mutations in most important transcription factor is MYC gene. MYC gene is commonly mutated in neuroblastoma. It's a very commonly mutated gene, neuroblastoma, diffuse large B lymphoma. In fact, if you see, MYC gene is responsible for signal transduction, transcription, protein biosynthesis, formation of microRNA, cell adhesion, others, and all those things. So MYK protectins is transiently induced by RAS MPK pathway, which we have discussed earlier. Its concentrations are well controlled, but if the MYC gene itself is mutated, it can lead to aberrant activation. And in fact, MYC gene mutations in fact leads to very aggressive carcinomas, especially seen in neuroblastomas and diffuse large B-cell, which usually called double hit or triple hit lymphomas. The functions of MYC mutants, MYC target gene cyclin D, which are directly involved in cell cycle regulation. It also upregulates the expression of ribosomal RNA and ribosomal RNA processing. MYC upregulates metabolic reprogramming and also Warburg effect. It increases the expression of telomerase and it reprograms somatic cells to form stem cells. It will cause de-differentiation. In fact, we can say there is some concept that we get introduced in metastasis, epithelial to mesenchymal transition. Mesenchymal cells are stem cells. They can exhibit more number of proteins. They have greater proliferative capability. They can migrate from one part to others. So MYC protein, though being an oncoprotein, also contributes to being a gene that is responsible for metastasis, also invasion, and it's also most importantly, it is all involved in reprogramming of a cell. So mutations are seen in Burkitt's lymphoma, neuroblastoma, also seen in breast, colon. So they are functionally identical. NMYC and LMYC genes also amplified in neuroblastoma and non-small cell lung cancer. Mutations in MYC involving components. So these mutations can happen upstream or downstream, but finally MYC protein is upregulated because MYC protein is responsible for many things. So we don't have this therapeutic window wherein we can produce an inhibitor or discover a chemical which selectively, even though MYC is inhibited, it causes a lot of side effects because it's also involved in a lot of normal physiological processes. So oncogenic mutations involving the components of upstream pathways elevate MYC proteins by increasing MYC transcription, enhancing MYC messenger RNA translation, or stabilizing MYC protein. Constitutional activity of RAS, MAP pathways in many cancer, NOT signaling, WNT signaling, and H-log signaling can transform the cells through the MYC upregulation media. There are certain single nucleotide polymorphism associated with inherited risk of prostate cancer and ovarian cancer. So what are the special note, inherited polymorphisms are they are just changes which are seen in many people along. It's not a mutation. It is the mutation is present both in the same person, but when you compare that mutation to general population, these people have a different kind of DNA sequence. So what is the difference between mutation and a polymorphism? Mutation is something, the same kind of DNA sequence is not even present in in this normal cell of the same person. But polymorphism, the same kind of DNA sequence is seen in the normal counterpart of the same individual, but it is not seen in the other individual. So polymorphism will give a selective advantage of that person to either in fact disadvantage to either develop cancer or to develop cancer or to not to develop cancer. So mutations in cell cycle regulators, growth factors, transduce signals that stimulate an orderly progress. So cell cycle is very regulated process. So there are something called cyclin decay kinases and cyclin kinase inhibitors. All these are tightly regulated. So that the tumor cycle actually happens in orderly factors. The regulators of cycle called cyclin dependent kinases, cyclin D1, cyclin dependent kinases, and cyclin dependent kinase inhibitors also contribute to normal cellular processes. The mutations in cyclin D1s or cyclin dependent kinase processes will also lead to formation of aberrant cells and aberrant cellular proliferation. Here is an example of mutation in cyclin D genes and CDK4. Gain of function mutations in cyclin D genes and CDK4 which promote unregulated G1S progression and thus function as oncogenes. There are three cyclin D genes, D1, D2, and D3, which are functionally interchangeable and often dysregulated by the acquired mutations of cancer cell. Application of cyclin D CDK4 is usually seen in melanomas, sarcomas, and glioblastomas. Examples is cyclin D1. Cyclin D is mutated. Cyclin D usually forms a complex that phosphorylates retinoblastoma, allowing the cells to retinoblastoma in hypophosphorylated form will stop G1S translation. CDK protein will actually phosphorylate RB and makes it inactivated. Thus, removing the inactivation will make the cell progress to G1 through G1S checkpoint. Other cell cycle inhibitors, which act as the tumor suppressor genes, may be also mutated and they lose their function, which may lead the cell to formation into cancers. The important is INK4, CDKN2AC mutations, P16 mutation, binds to cyclin D and promotes the inhibitory effects of RB. The mutation, so P16 ink actually facilitates RB in inhibiting the cell cycle. Mutation in this one will make the RB weak, and because retinoblastoma gene becomes weak, if the cell cycle is progressed. So as told earlier, sometimes inhibitors of we have intrinsic inhibitors of cell cycle progression, CDKDNA1. The mutations in CDKDNA1 also will contribute to the most important example, germline mutations of P16 CDKNA. They are present in 25% of melanoma prone. In fact, familial melanoma syndrome is also is due to mutations in the CDKN2A, or which is usually called the P6. P16 is the protein, the CDK2N is a gene. Due to the mutations in CDK can cause familial melanoma syndrome. Promise me that you will never always remember that you are braver than you believe. All these quotes are actually all intended for the cancer patients. I wanted someone might get inspired by this lecture series, take up oncology, and with passion they may serve people, and all these things should reach the cancer patient on my behalf. Thank you very much.